Hello everyone, my name is Justin Serio, and today I'm going to be talking to you guys about patterns. Specifically though, I'm going to be talking to you about how patterns are being utilized in the fight against cancer to help improve outcomes for the disease and help patients. So to start off, I want everyone to kind of think back to about 100 years in the past and think about how health was substantially different then than it is now. What kinds of things did we deal with then in terms of disease that now we might have cures for, and vice versa. So the thing I usually think about is a game I played as a kid, which is Oregon Trail. And essentially in that game, you kind of pick a group of people to go out and venture into the frontier. You ford a couple rivers, hunt a few rabbits, and then everyone invariably dies of dysentery or cholera. And it's really bad and you lose and it's super frustrating. You have to start all the way back at the beginning. But even though this is a game, that wasn't far from the truth in the real world at that time. So if we look at this graph showing the causes of death in 1900 in the United States, you can see that diseases like cholera that were infectious diseases made up over 50% of the things people died from at that time. And then over time, if we compare that to today, things are in fact very different. Whereas we see in 2010, the infectious disease part of the pie, or the green part of the pie, is essentially completely gone due to medical advancement. However, despite things improving for that particular case, things have gotten worse for other things. So I want to draw your attention to the blue part of the pie, which is cancer, which is now becoming quite a huge problem in medicine today. So at first, I kind of thought, okay, well, maybe cancer's been around in our kind of awareness for much less time than these infectious diseases like cholera. And maybe that's why we haven't figured out how to solve it. However, looking this up, I was surprised to find that cancer has been first recorded in ancient Egypt and was named in ancient Greece, so it's been around for thousands of years. And on top of that, we get all the way until the 1940s before we even have the first chemotherapy or the first drug that effectively treats cancer. And if we go 75 years to the present day now, cancer's still around. So what's going on? It's not even hard for someone to hold this opinion, which is essentially, is cancer super hard, or are people working on it just really dumb? And hopefully I can convince you that it is indeed a very difficult disease to work with, and we do kind of have a very good idea of what we're doing and how to fix it. So first of all, if you want to solve cancer, you have to know what it is, right? So what is cancer? Quite simply, it comes about from a damaging event to a normal healthy cell in your body, and this changes it into a cancer cell. It then grows and grows and grows until it becomes a large mass of cells, which we call tumors, which people have usually heard about. And when you get these tumors in different organs of your body, they can cause problems for the organ to function, leading to them to fail, and then ultimately making you sick. So this is pretty much what cancer is, but how exactly does it happen? So quite simply, cancer stems about from a molecule many people know about called DNA. And DNA is the instruction booklet of life. It tells your cells what to do when they need to do it and how to do it. And the way it does this easily is by subdividing the instructions into subsets called genes. And each gene corresponds to a particular process or function the cell needs. So one gene may convey to the cell how to move, while another gene may convey to the cell how to divide if you need to repair a wound or if you need to develop from a baby into an adult. So DNA, like I said, is the main reason cancer comes about, but it happens to be very fragile. So your healthy DNA is kind of sitting there and it can be damaged by many things despite the cell trying to help protect it. These things include things like solar radiation from the sun, a long list of chemicals called carcinogens or cancer-causing agents, as well as viruses. And when these interact with your DNA, they can lead to permanent changes to it, which we call mutations. And if these mutations occur, they can ultimately produce cancer. These are the driving force of what cancer comes about from. So what else can happen if you get a mutation? So like I mentioned, cancer can be a very common cause of what happens after mutation. However, some mutations do almost nothing at all, and this makes it difficult to evaluate them from a therapeutic standpoint. Additionally, some genes can be so incredibly important that if they're mutated, the cell just can't survive at all, whether it turned into a cancer or whether it's normal and healthy, and the cell will die. So you can kind of think of this as a balancing act, right? So you have all these genes, which are represented as blocks here in this figure, and you can imagine that they're constantly trying to maintain a state of normal action. So they want to keep the cell healthy, keep the body healthy. And if some of these become mutated, like I've shown below, they kind of shift how the balance sits. And you could imagine if enough mutations are acquired, the balance shifts toward cancer and the cell changes and causes tumors and hurts your body. Whereas the opposite can happen as well, where it shifts at very important genes and ends up damaging your cell so badly that it dies, which isn't good as well. So if we kind of expand on the problem of why is cancer so complicated, it's due to the fact of many factors, which I'll list here. So one of these factors is that you don't just have a couple gene blocks like I showed in the last figure. You actually have 25,000 genes in each of your cells, which is a lot to handle. So that's kind of one variable. There's 25,000 genes to keep track of. On top of that, you could imagine that genes have different functionality over time. So if a gene needs to be working to tell the cell how to move, for instance, it might display as green. If it needs to be off, like when a cell is trying to conserve energy, it may not want to actively seek energy out. It may be red. So with these gene patterns, they're also another variable to take into account. We have a lot of genes, but they're also doing different things. And on top of that, like I mentioned before, cancers can occur in a wide array of places. So you have different cancer types like brain or lung or blood, and those all look and act differently. And even things that are in the same organ but in different locations can additionally add some complexity to how to treat them and how they'll behave. So if we take each of these 25,000 genes and put them together, we get a cube. And in this case, let's imagine it like a puzzle or a Rubik's Cube, where in a normal state, all of these 25,000 genes are organized like a solved Rubik's Cube. And when we get enough mutations, the cube starts to get jumbled up, just like a Rubik's Cube we're playing with as a kid. And our job is to basically put the cube back together as we found it to make the patient healthy again. But this is only a representation on the screen here. It's 17 by 17. But to represent all 25,000 genes in your cell, it would have to be 4 times the volume, or a 65 by 65 sided Rubik's Cube. That is humongous. And on top of that, everybody's DNA is inherently different from person to person. So if 2 patients with the exact same cancer come into the clinic, the solution for their Rubik's Cube might be different, further complicating curing both patients with the same treatment. So the way I've set this up makes it seem impossible. How are we going to solve this Rubik's Cube? It's ridiculously big. There's too many parameters to keep track of. I can't even solve a 3x3 Rubik's Cube myself. What are we going to do? Well, luckily for us, recent medical technologies have allowed us to get at this by looking at the patterns of genes. So this technology here shown is a heat map, and this allows us to do just that. We can look at the patterning of genes in different types of cells. So in this case, genes are in the rows and patient type is in the columns. And we can see here that if you have a red gene set on top and a green gene set on the bottom, that conveys to a normal patient or a healthy patient, whereas the cancer patients tend to have the opposite pattern. This allows us to make kind of very specific decisions about what to do with each patient based on their gene pattern. And although this is a simple example, these can be much more complicated as shown here. But despite this complexity, we can still note patterns and make good decisions. So again, on this case, the patients on the top have red on the left, green on the right, while as the patients on the bottom tend to be different and they have green on the left and red on the right. So we can still interpret the patterns despite the complexity. So let's imagine we take just we want to identify the difference between cancer and a normal cell. So we look at the pattern, we see that, oh, these 2 genes, for example, happen to be very different in the cancer cell than a normal cell. So what we can do is design a drug that'll target those genes in both cells. And ideally what will happen is the normal cell, those genes aren't that important for it to survive, and it stays alive. Whereas in the cancer cell, those genes are extremely important and are what makes it a cancer. And if those genes are targeted by a drug, they'll die, killing the cancer cells. This would be a huge improvement upon current therapies, which don't distinguish between healthy and normal cells and kill everything. And that's why we have such large side effects. So if we can target specifically cancer, this can drastically help patients. Another thing we can do is to look at patterns and kind of make broad conclusions from them. So if a patient comes into the clinic and we see that if they have this particular pattern and we give them treatment A, they tend to do substantially better than if we give them treatment B or C. And this is pretty much personalized medicine. Patient comes in with a specific pattern, we give them a specific treatment, they have the best possible outcome for them. Another thing we can do is more of a predictive modeling approach, where we can say before cancer even happens, if we see this pattern, it corresponds to patients that will develop cancer soon. Or if they get a cancer, it's going to be really severe and need to be treated much differently. So doctors can look at that information and say, you're gonna get a cancer at this particular time, and we're gonna need this particular treatment regimen to help you specifically. So to swing back to Oregon Trail, like I mentioned before, in those times they had no idea what was going on with cholera, killing over 50% of people, along with other infectious diseases at the time, and they had no idea how to solve that problem. And although we've reduced that number from 50% to almost zero with medical technologies, cancer is creeping up on the causes of death lists today. But now, due to technologies, we're able to frame cancer as a big Rubik's Cube puzzle of genes that can be manipulated. We can now identify the patterns that those genes make, allowing us to separate patients into groups, allowing us to reduce the cost of medical treatment, identify which patients need what treatment and when, allowing us to reduce side effects, reduce costs, and improve the outcomes for patients so they live longer and healthier lives.